Let us continue our discussion about regulation of gene expression by molecules which bind to cell surface receptors and binding of these molecules to the cell surface receptors ultimately through specific signal transduction pathways lead to the activation of specific genes which ultimately manifest into distinct physiological responses. Now, in the last 5 classes we have been discussed about these kind of signal transduction pathways. I am trying to give you an overall picture of how these signaling molecules which interact with specific membrane receptors can transduce signals through various signal transduction pathways involving specific protein kinases and ultimately leading to the activation of specific transcription factors and activation of specific target genes. So, just to recapitulate what we have, so today what we will discuss is how this kind of a regulation of gene expression takes place by a distinct group of molecule called cytokines. So, this will be the today's topic. In the previous 2 topics we have discussed about signaling molecules which activate transcription through G protein coupled receptors. We also discussed molecules which like growth factors which bind to growth factor receptors and then activate transcription through the RAS pathway leading to activation of specific genes. So, just to recapitulate what we have been discussing, I have just summarized here almost what we have been discussing in the last 4 lectures. We began by discussing a group of molecules, signaling molecules which interact with specific membrane receptors known as GPCRs or the G protein coupled receptors, which are 7 transmembrane proteins, and when these signaling molecules interact with the GPCRs, it results in the activation of G proteins, and we have discussed about what kind of G proteins are these. These are trimeric G proteins which consists of alpha, beta and gamma subunits, and when the molecule interacts with the receptor GPCR, it alters the conformation of the receptor and resulting in the activation of the G protein, and the alpha subunit of G protein now dissociates, and this alpha G subunit which is an active GTPase now activates adenylate cyclase. Adenylate cyclase then synthesizes cyclic AMP, which is a very important second messenger. Cyclic AMP in turn activates protein kinase A. We also discussed how the same in certain cases when certain signal molecules dock with GPCRs, it can also lead to different kind of G proteins, and these kinds of G proteins instead of activating adenylate cyclase, they can actually activate phospholipase C, and this activated phospholipase C now converts the 1,3-bis inositol diphosphate into inositol triphosphate and diacylglycerol. Now, diacylglycerol then goes and activates protein kinase C, whereas the inositol triphosphate goes to endoplasmic reticulum, releases calcium. Calcium has 2 things: it can either go and activate calmodulin, and this in turn can activate calmodulin kinases, or the calcium can also go along with diacylglycerol can activate protein kinase C. So, this is what we discussed about the GPCRs. We also discussed in detail how certain growth factors, polypeptide growth factor like epidermal growth factor, fibroblast growth factor, platelet derived growth factors, etc., how when they interact with specific membrane receptors known as receptor tyrosine kinases, the binding of the signaling molecules with these receptors activates the tyrosine kinase activity of this receptor, and these 2 monomers cross phosphorylate each other on the tyrosine residues, and these tyrosine phosphorylated receptors now interact with specific adapter proteins like the GRB2, which then activates the GTA guanine nucleotide exchange factor, and then which activates the RAS. RAS is a monomeric GTPase which is very similar to the G alpha subunit of the trimeric G proteins, and this activated RAS then activates what is known as the MAP kinase signaling pathway involving MAP kinase, which then activates MAP kinase, and then it activates MAP kinase. There are also certain things which we did not discuss because it is not possible to cover all the signal transduction pathways. The same RAS which has been activated by the growth factors can also go and activate a PI3 kinase, phosphatidylinositol 3 kinase. This ultimately leads to a different kind of signaling pathway, and this can lead to the activation of protein kinase B. So, what we have discussed so far is that different molecules interacting through different signaling pathways can lead to the activation of different protein kinases. In one case protein kinase A is activated, in another case calmodulin kinase is activated, or protein kinase C, MAP kinases, or protein kinase B. Now, what is the purpose of all these things? And ultimately all these kinases go and phosphorylate a number of target proteins which varies from cell type to cell type depending upon the physiological situation as well as crosstalk with other signaling pathways, and these target proteins can be either metabolic enzymes, it could be ion channel proteins, or it could be transcription factors. In this lecture series, we focus primarily our attention on how all the signaling pathways lead to the activation of transcription factors, and once these transcription factors are activated, it ultimately results in the activation or repression of specific target genes, which in turn manifests into distinct physiological responses. So, this is what we discussed in the last 5 classes. Today, we will talk about another important group of molecules other than those the G protein ligands as well as the receptor tyrosine kinases. Let us now talk about a new set of molecules called cytokines. How cytokines regulate gene expression? This is what we are going to discuss today. Now, cytokines are proteins produced by diverse cell types, and they primarily interact with cells of immune system in order to regulate the body's response to disease and infection. So, the cytokines play a very important role in the way we fight various infections, whether it is viral infections, bacterial infections, parasite infections. These cytokines play a very important role in modulating our immune functions. The cytokines also regulate several normal cellular processes in the body. So, they are very important biological molecules. Now, what are the various types of cytokines? Cytokines are very diverse group of molecules. They include colony stimulating factors, many of these like GCSF, GM-CSF, etc. They stimulate the production of blood cells such as red blood cells, platelets, and so on and so forth. There are also cytokines also involve growth and differentiation factors. They function primarily during development. When we studied the importance of regulation of gene expression during development, we will come back and revisit some of these molecules. The cytokines also are immunoregulatory and pro-inflammatory cytokines, and these include very important molecules such as interferons, interleukins, tumor necrosis factor alpha. All these molecules play a very important role in regulating the function of the immune system, especially the T cells, B cells, and so on and so forth. How do the cytokines work? Cytokines are actually released by various cell types into the circulation or sometimes directly into a particular tissue, and once the cytokines are released, these cytokines then go and interact with specific target cells, and this interaction primarily takes place through specific cell surface receptors which are present on the target immune cells. So, there are cells which produce these cytokines, these are released into the bloodstream, and these cytokines then go and bind to the cell surface receptors of specific target cells, and this cytokine receptor interaction then activates specific signal transduction pathways, leading to the modulation of activity of a number of proteins, which also includes transcription factors. Ultimately, all these things manifest in the form of distinct physiological response. So, this is molecules like interferons, interleukins, erythropoietin, growth hormones. All these molecules primarily act through this particular signaling pathway. Of course, this is a signaling pathway we have also discussed in the previous lectures. This is the same mechanism by which other signaling molecules also activate expression of specific target genes. But let us now discuss what is the distinction between the way the cytokines activate specific signal transduction pathways and molecules like growth factors and other G protein coupled receptor mediated pathways are different. Now, before we discuss the mechanism by which the signal transduction pathway takes place, let us now briefly discuss what are the various kinds of cytokines and what is their importance. The reason we need to understand is because unless we know the importance of the cytokines, there is no point in discussing about these various signal transduction pathways. So, what I have listed in the last couple of slides is to variously introduce what are the very important cytokines and what are the normal functions, and then it becomes very clear why we need to understand the mechanism by which these cytokines work. For example, there are a whole bunch of molecules known as interleukins, which form a very important group of cytokines, and there are very diverse type of interleukins. I have listed some of these and also discussed the functions of some of these cytokines. For example, you have interleukin 1. Interleukin 1 primarily activates antigen presenting cells as well as CD4 positive lymphocytes after the differentiation of B cells and T cells. They also affect the differentiation of B cells and T cells as well as many immunocompetent cells. And they also take part in the regulation and production of various other cytokines like the granulocyte macrophage colony stimulating factors or GM-CF. And remember, molecules like interleukins and GMs are very important biological molecules, have a lot of biomedical importance in the treatment of a variety of diseases. Similarly, interleukin 2 stimulates the proliferation and activation of B cells and T cells, and it plays a very important role in the differentiation of T helper type Th2, T helper 2 T cells, as well as in allergic responses and switching of various antibody types. If you now take interleukin 5 primarily stimulates the production and maturation of eosinophil during inflammatory responses, whereas interleukin 8 acts as a chemotactic factor that attracts neutrophils, basophils and T cells to the sites of inflammation. You can see these molecules are very important in many of these processes. Similarly, interleukin 12 is a very critical linker between innate immunity and adaptive immunity, capable of Th1 differentiation and interferon gamma released by T cells as well as natural killer cells. So, all these interleukins play a very important role and regulate a number of important biological responses. Similarly, if you look at interleukin 10 acts to repress the secretion of pro-inflammatory cytokines. So, it suppresses inflammation, it is an anti-inflammatory factor. Interleukin 3 is a potent activator of hematopoietic cells. It stimulates natural killer cells and also acts as in synergy with IL-4 during the induction of CD4 positive lymphocyte activation process. Interleukin 7 is known as a growth factor of the immature B cells and T cells. It induces apoptosis of tumor cells and causes differentiation of cells from a subgroup of acute myeloblastic leukemia, a very important and difficult form of cancer. Interleukin 9 stimulates the excretion of IL-2, IL-4, IL-6 and IL-11 and takes part in a stimulation of cytotoxicity of T cells, T killer cells and natural killer cells, inducing apoptosis. Whereas interleukin 11 is a pro-inflammatory factor and it regulates the functions of B cells as well as T cells, and it also takes part in the induction of various killer cells activities and acts as an autocrine factor for the proliferation of megakaryocytes. So, you can see this diverse group of interleukins play a very important role in a number of biological processes. So, it is very important for us to understand how do these molecules are able to bring out these distinct physiological responses. Similarly, if you now take another group of cytokines like TNF alpha and TNF beta, the secretion of these TNF alpha and TNF beta, TNF stands for tumor necrosis factor. There are two types, TNF alpha and TNF beta. These are secreted by the Th1 helper cells and they activate macrophages, they inhibit apoptosis of neutrophils and eosinophils and induce vascular endothelial cells at the sites of infection to change the addition molecules they express, so that phagocytes circulating the blood can bind to them. So, has a very important role in immunosurveillance during infection as well as disease processes. Similarly, interferons, which consists of interferon alpha, interferon beta and interferon gamma, they are produced in the area of infection during very early stages of immune response. Those who study immunology understand that these molecules are very important for the immune functions. Interferon alpha and interferon beta, for example, they induce proliferation of natural killer cells and stimulate innate as well as adaptive immune responses, and they specifically targeted for virus infections. So, the interferons play a very important role in our ability of our body to control various viral infections. Upon activation, the natural killer cells actually release interferon gamma, which activate macrophages to secrete various cytokines, and these cytokines in turn activate macrophages to secrete cytokines that help activate T cells and promote initiation of specific antigen specific T cell responses. So, you can see these molecules play very important in both humoral as well as cell mediated immune responses. So, to put in a nutshell, the cytokines are key regulators of various immune functions. For example, they alter gene expression. This is what we are going to study. How these cytokines, by interacting with cell surface receptors, how they alter gene expression is what is going to be the focus of today's talk. How do they alter gene expression? How do they promote growth? They either boost interleukin 2, these cytokines either boost or suppress immune responses. For example, if you take molecules like interleukin 2, they actually boost immune responses, whereas molecules like IL-10 and TGF beta 1 suppress the immune responses. So, they are both positive as well as negative regulators of immune responses. Whereas type 1 interferons and tumor necrosis factor alpha are mediators of natural as well as innate immunity. Whereas the various interleukins listed here, they are regulators of lymphocyte growth, activation and differentiation, and interferon gamma is an activator of inflammatory cells, whereas interleukin 3, GM-CSF and IL-7 stimulate hematopoiesis like production of red blood cells, platelets and so on and so forth. So, I have spent all this time to primarily bring to your attention that the molecules or the signal transduction pathways we are going to discuss are very important, and these molecules play a very important role in a number of immune functions. So, we need to understand how do these molecules act and how do they transduce signals once they bind to distinct cell surface receptors. The other important reason why we have to know how these molecules act is that when these cytokines which were discussed so far, when these cytokines are overproduced in the body, or when they are not produced properly, or when the production is not properly regulated, it can lead to number of disease processes. For example, overproduction or inappropriate production of certain cytokines, it can lead to number of diseases. The example is in the case of rheumatoid arthritis, for example, cytokines such as interleukin 1 as well as tumor necrosis factor alpha are produced in excess, and when these cytokines are produced in excess, it leads to rheumatoid arthritis, inflammation of joints and so on and so forth. So, a number of companies have either brought out or in the process of developing molecules which can suppress the production of these interleukins, so that you can reduce inflammation and you can find a cure for rheumatoid arthritis. Similarly, interleukin 4, when it is produced hormones, it causes allergy involved in allergy and allergic diseases, whereas interleukin 17 is involved in a number of autoimmune disorders. This is just a few examples to tell you many of these molecules are very important, and if they are not properly produced or if the regulation is not proper, it can manifest in the form of specific disease processes. So, in view of the importance of these molecules in various biological processes as well as its importance in controlling these signaling molecules, a number of drug companies have invested a lot of money to understand the mechanism by which these cytokines act, and in the process have discovered a number of drugs which either activate or act as inhibitors of these various cytokines, and many of these drugs are already in the market. So, a number of drugs have been developed which act by inhibiting the action of cytokines. I just lifted a few examples here, and towards the end of this lecture we will again revisit this and look at various other drugs which have been developed for various diseases. For example, you have a drug called Kineret, or also known as anakinra, which is used in the treatment of rheumatoid arthritis. It basically works by inhibiting interleukin 1 binding to the receptor. So, it is an antagonist for interleukin 1. When it goes and binds to interleukin receptor, it prevents binding of the interleukin 1, and as a result the entire signal transduction pathway leading to gene expression is blocked, and this has a very important effect. So, these kind of inflammatory cytokines, the inflammation is now reduced, therefore, it is very widely used for rheumatoid arthritis. Similarly, molecules like Enbrel, Remicade, Humira, they are all very important drugs. They are all TNF alpha inhibitors. They are also popularly known as TNF blockers. So, they all block the TNF mediated signal transduction pathway. They basically bind TNF and prevent TNF from binding to the cell surface receptors. So, you can see by blocking these molecules from interacting with the cell surface receptor, you can develop a number of therapeutic agents for specific diseases, whether it can be rheumatoid arthritis, inflammation, or a number of other disease processes. So, it is very important for us to understand what happens in the normal process and what kind of signal transduction pathways are blocked when you block the interaction of these molecules with the cell surface receptors. So, having discussed so far the importance of these cytokines, how they control a number of important biological processes, right from production of erythropoiesis or various blood cell types as well as immune system functions. Now, let us try to understand how do the cytokines are able to bring out these distinct physiological responses. So, the first step in the cytokine action is their binding to cytokine receptors. So, this is the first step in the cytokine signal transduction pathway. So, let us now try to understand what kind of cytokine receptors interact with these specific cytokines we discussed so far and so forth. Now, there are three distinct types of cytokine receptors. These are classified as type 1 receptors, type 2 receptors, and soluble receptors. Now, the examples for the type 1 cytokine receptors are type 1 interleukin receptors, erythropoietin receptors, granulocyte macrophage colony stimulating factor, GM-CSF receptor, granulocyte colony stimulating factor, GCSF receptor, growth hormone receptor, prolactin receptor, oncostatin M receptor, and leukemia inhibitory factor receptor. So, all very important receptors. They bind to very important ligands which has very important roles. So, all these receptors belong to the type 1 cytokine receptors. The type 2 cytokine receptors, some of the examples are the type 2 interleukin receptors, interferon alpha as well as interferon beta receptor as well as interferon gamma receptor. In addition to this, you have other types, those belong to the immunoglobulin superfamily, which include the interleukin 1 receptor, colony stimulating factor 1, CSF1, c-KIT receptor, the interleukin 18 receptor. And you have what is called as the tumor necrosis receptor family, which involves CD27, CD30, CD40, CD120, and lymphotoxin beta receptor. And we also have what are called as chemokine receptors, which play very important role in infections such as HIV, and HIV in fact requires many of these chemokine receptors, especially the CXCR4 acts as a co-receptor for HIV1, and without this receptor the HIV cannot transduce the signal further. Some of the chemokine receptors include interleukin 8 receptor, CCR1, CXCR4, MCF receptor, NAP2 receptors. And finally we have these TGF beta receptors, which include TGF beta receptor 1, TGF beta receptor 2. So the cytokine receptors are of diverse cell types, and some of these various cell types we just discussed now. Now what are the distinction between these type 1 receptors and type 2 receptors? The type 1 cytokine receptors are basically transmembrane receptors expressed on the surface of cells that recognize and respond to cytokines with 4 alpha helical strands. So cytokine molecules which interact with the type 1 receptors contain 4 alpha helixes. They share a common amino acid motif called WSXWS in the extracellular portion of the adjacent to the cell membrane. So I have just listed here, this is the extracellular domain of the cytokine receptor, and this red one is the intercellular domain, and you can see just the region adjacent to the cell where it binds the cell membrane, you have this WSXWS motif, which is a signature motif for all the type 1 cytokine receptors. The members of the type 1 cytokine receptor family comprise different chains, some of which are involved in the ligand cytokine receptors and others are involved in signal transduction, and in the far end of this extracellular domain they also contain a number of conserved cysteine residues. So these two, presence of conserved cysteine residues as well as presence of the WSXWS motif, is a very characteristic feature of this type 1 cytokine receptors. The type 2 cytokine receptors are also transmembrane proteins. They are also expressed on the surface of cell types, and they bind and respond to select group of cytokines, which we discussed earlier. And these receptors are similar to the type 1 cytokine receptor, except that they do not contain the signature motif which we saw in the type 1 receptors, namely the WSXWS motif, which is very characteristic of the type 1 cytokine receptors, is absent in the type 2 receptors. The other important thing about the type 2 receptors is that the intracellular domain of the type 2 cytokine receptors typically associates with a tyrosine kinase belonging to the JNS kinase family, or popularly known as the JAK family. The JAK kinases are the JNS kinases. I have just listed here the various cytokine receptors which belong to the type 1 cytokine receptors. A number of interleukin receptors belong to the type 1 receptor, and the type 2 receptors, again certain interferon receptors, both alpha, beta and gamma receptors, as well as certain types of interleukin receptors belong to the type 2. In addition, we have the colony stimulating factor receptors such as the erythropoietin receptor, GM-CSF and GCSF, and there are various other receptors which are also classified as cytokine receptors, which include the growth hormone receptor, prolactin receptor, oncostatin M receptor and leukemia inhibitor factor receptor. So these are the cytokine receptor family that and the mechanism that we are going to discuss today. All these receptors primarily act through the signal transduction mechanism that we are going to discuss in the next few minutes. So we have discussed so far what are the various cytokines, what are the importance of the cytokines, what are the functions of the cytokines, and we also discussed what is the first step in the cytokine action, namely they go and bind to specific cell surface receptors which are of different types like type 1, type 2 and so on and so forth. Now ask the question, once the cytokine binds to the receptor, what happens next? Once the cytokine binds to the cytokine receptor, then it initiates a specific signal transduction pathway. So let us now understand what is the cytokine signal transduction pathway, what exactly happens once the cytokine binds to a cytokine receptor. Now the signal transduction pathway transduced by cytokines and cytokine receptors are popularly known as the JAK-STAT pathway. Now JAK stands for JNS kinases. These are the kinases which are the primary molecules which are attracted to the cell surface receptors once the receptor binds to a cytokine, and the STAT refers to signal transducers and activators of transcription. These are very unique kind of transcription factors that is very unique for the cytokine signaling pathway. So let us now go in detail and understand what exactly is this JAK-STAT pathway. Now the important distinction that we have to remember before we go into the details is that the cytokine receptor that we have discussed so far of the type 1, type 2 and various other families of cytokine receptors, they are very different from the receptor tyrosine kinases which we discussed in the previous class, which are the growth factor receptors, in that the growth factor receptors possess intrinsic tyrosine kinase activity, whereas the cytokine receptor do not contain any tyrosine kinase activity. In fact, they do not have any protein kinase activity. So, this is one important difference between the cytokine receptors and the receptor tyrosine kinases or growth factor receptors. Growth factor receptors are intrinsic tyrosine kinases, whereas cytokine receptor do not have any kinase activity. Now, then how do they transduce the signal? What happens once the cytokines bind to the receptor, they interact with members of a very unique family of cytoplasmic protein kinases known as the JNS kinases or JAKs. So, in the case of growth factor receptors, when the growth factor binds to the growth factor receptor, the tyrosine kinase activity of the receptor itself is activated. So, the two monomers cross phosphorylate each other on the various tyrosine residues, and this results in the activation of signaling transduction pathway. Whereas, in the case of cytokine signaling, when a cytokine binds to a cytokine receptor, it actually uses the recruitment of specific tyrosine kinases to the receptor, and these kinases are called as JAKs or JNS kinases. Now, once the JAKs physically associate with the membrane region, this is what I have shown here. Here is the cytokine, here is the receptor, and here are the JAKs. So, once the JAKs physically associate with the membrane proximal region, that is the region of the receptor very close to the cell membrane, this results in the tyrosine phosphorylation. The JAKs cross phosphorylate each other, and also they phosphorylate the tyrosine residues of the cytokine receptor, which is shown in the next slide. So, once the cytokine binds to the cytokine receptor, it recruits the JNS kinases now are activated, and these JNS kinases now phosphorylate the cytoplasmic domain of the cytokine receptor. And then what happens? The activated JAKs phosphorylate the receptors as well as the cytoplasmic proteins belonging to a family of transcription factors called signal transducers and activators of transcription. So, once the JAKs phosphorylate the cytoplasmic domain of the cytokine receptor, this phosphorylated receptor now interacts with specific molecules known as the signal transducers and activators of transcription, or abbreviated as STATs. So, these STATs are very unique group of transcription factors which are very unique and characteristics of the cytokine signaling pathway. So, the JAKs phosphorylate the cytoplasmic domain of the cytokine receptor, and this phosphorylated receptor now acts as a docking site for the STATs, and once the STATs go and bind to the phosphorylated tyrosines of this receptor, these STATs are again phosphorylated by the JAKs, and then these phosphorylated STATs now form either homodimers or heterodimers, and then they go into the nucleus, bind to distinct DNA elements or the enhancer elements, and then activate transcription of specific genes. So, you can see this cytokine signaling signal transduction pathway is quite different from the GPR signaling pathway and the receptor tyrosine signaling pathway, which we discussed earlier. So, here you have a introduction of a very new mechanism involving what are called as JNS kinases, which are cytoplasmic tyrosine kinases, and another important group of molecules known as signal transducers and activators of transcription or STATs. So, to emphasize this point, what I mentioned, I just put a cartoon here just to tell you the two important distinctions between a growth factor signaling pathway and the cytokine signaling pathway. In the case of the growth factor signaling pathway, when the growth factor binds to the growth hormone receptor, growth factor receptor, the receptor itself is a tyrosine kinase. It results in the autophosphorylation of the tyrosine residues on the surface of the receptor tyrosine kinase, and results in the activation of the receptor tyrosine kinase. And these phosphorylated tyrosines now act as a docking site for proteins like GRB2. And then through SOS, the RAS is activated, and then MAP kinase pathway is activated, and transcription factors are phosphorylated, leading to activation of genes. So, here the receptor itself is a tyrosine kinase. Whereas, in the case of cytokine signaling pathway or the JAK-STAT signaling pathway, when the cytokine binds to the receptor, it actually the intracellular domain of the receptor actually serves as a recruitment site or a docking site for tyrosine kinases, and the receptor itself is not a tyrosine kinase. Here the receptor is a tyrosine kinase, here the receptor only serves as a docking site for a cytoplasmic tyrosine kinase. So, here when the hormone binds the receptor becomes the tyrosine kinase, whereas in the case of cytokine signaling, when the cytokine binds to the cytokine receptor becomes the docking site for a cytoplasmic tyrosine kinase known as the JNS kinases or the JAKs. So, these are the two important differences between a receptor tyrosine kinase signaling pathway or the growth factor signaling pathway and the cytokine signaling pathway. Now, so we have now studied about cytokines, we understood various cytokine receptors, and then we told that once a cytokine binds to cytokine receptor, it results in the recruitment of a specific group of kinases known as the JAKs or JNS kinases. and this JNS kinases then phosphorylate the receptor that results in the recruitment of the STATs. So, let us now spend some minutes to understand what are these JNS kinases and what are these STATs or the signal transducers and activators of transcription. Now, the JNS kinases or the JAKs belong to a family of non-receptor protein tyrosine kinases. Important, this term is very important, where the receptor tyrosine kinases or RTKs are important transmembrane receptors for growth factor signaling, whereas the JNS kinases are different from those, so they are called as non-receptor protein tyrosine kinases. In fact, they are referred to as the cytoplasmic tyrosine kinases, which are recruited to the transmembrane receptor or the cytokine receptor. So, the JAKs belong to a family of non-receptor tyrosine kinases of approximately 130 kilodalton in size, and they basically comprise of 4 different kinases known as JAK1, JAK2 and JAK3, as well as TYK2 known as the non-receptor protein tyrosine kinase 2. So, JAK1, JAK2, JAK3 and TYK2, these are the 4 kinases or 4 non-receptor cytoplasmic tyrosine kinases which interact with the cytokine receptors when these cytokine receptor interact with the cytokine molecule at the extracellular domain. And what do these JAKs do? The primary function of these JAKs is to phosphorylate the cytokine receptor, and once the cytokine receptor is phosphorylated, the STATs are recruited to the phosphorylated cytokine receptor, and then the JAKs phosphorylate the STATs also. And phosphorylation of these STATs are very, very important for the dimerization and then their nuclear translocation and activation of gene expression. Now, the STATs, there are about 7 different STAT molecules which have been discussed in mammals. These are known as the STAT 1, 2, 3, 4, 5, 6, STAT 1 to 6. This also includes STAT 5A and STAT 5B, which are encoded by distinct genes. So, there are about 7 different STAT molecules. Now, so in this particular slide, I have basically listed what kind of cytokine receptors interact with what kind of JNS kinases, and ultimately what kind of STAT molecules are activated. So, if you now take interferon gamma for example, interferon gamma, when interferon gamma interacts with interferon gamma receptor, it results in the activation of JAK1 and JAK2, which then phosphorylates interferon gamma receptor and recruits STAT1 to the receptor, and then STAT1 gets phosphorylated, and then STAT1 either forms homodimers or heterodimers with STAT2 results, and then goes into the nucleus and activates expression of specific genes. Whereas in the case of interferon alpha and beta, instead of JAK1 and JAK2, JAK1 and TYK2 form or activated, and these JNS kinases then come and activate STAT2. In the case of interleukin 2, JAK1 and JAK3 are the JNS kinases which interact with the IL-2 receptor, and they activate STAT5. Whereas in the case of interleukin 3, JAK2 is activated and JAK2 then goes and phosphorylates STAT5. IL-4 receptor primarily interacts with JAK1 and JAK3, and JAK1 and JAK3 then activates STAT6. Whereas IL-6, JAK1, sometimes other JNS kinases are also activated, but primarily JAK1 is activated, which then goes and phosphorylates STAT3, and this in turn goes to the nucleus and activates the expression of various genes. Whereas IL-10 receptor serves as the docking site for JAK1 and TYK2, which then activates STAT3, whereas in the case of IL-12, JAK2 and TYK2 are involved, they then activate STAT4. So, one can ask the question, why do you need so many STATs? They are all the same or they have different functions? The answer to this is in the next slide. Yeah, you can see each one of the STATs I have discussed so far, namely STAT 1 to 6 and STAT 5 and STAT B, each one have a very, very important function. So, then the organism does not have all these STATs for the same function, but each one of them have a distinct function. Now, how do we demonstrate that these STAT molecules have different functions? The best thing to understand the function of a particular transcription factor is you create a knockout mice. So, once you knock out this transcription factor, then you ask the question, what happens to the mice? So, if these STATs have redundant functions, then if you knock out one factor, the other factor will take over its function, so you should still be normal. But if these STATs have non-redundant functions, then you should get specific phenotypes, and that is what happens. You can see when you have STAT1 is knocked out, and these mice have interferon signaling. So, interferon signaling is drastically affected when you knock out STAT1, clearly indicating that STAT1 is very, very important for the interferon signaling. When you knock out STAT4, IL-12 signaling is drastically affected. Nothing happens to interferon signaling, clearly indicating that these two have different signaling functions, or they are involved in different signal transduction pathways. Similarly, if you knock down STAT5A, prolactin signaling is affected, indicating that when prolactin binds to prolactin receptor, it results in the recruitment of STAT5, and all the physiological effects of prolactin is actually manifested or is propagated by the STAT5 transcription factor. STAT5b knockouts have impaired growth hormone signaling. Similarly, STAT6 knockout have impaired IL4 and IL13 signaling. So, this basically tells you that each one of these STAT molecules go and activate different set of target genes, and that is why you have different physiological responses in response to the initial signaling molecule through which they got activated. Just to give you an example here of the various things we have discussed so far, if you take for example interferon alpha signaling, interferon alpha goes and binds to interferon alpha receptors, and when interferon alpha binds to interferon alpha receptor, it results in the activation of the JAK1 and TYK2, and these Janus kinases then phosphorylate the interferon receptor, and when the interferon alpha receptor is phosphorylated, that becomes the docking site for STAT1 and STAT2, and these again get phosphorylated by JAK1 and TYK2, and these phosphorylated STAT1 and STAT2 now form heterodimers, then they go into the nucleus and activate distinct set of genes. Whereas in the case of interferon gamma signaling, when interferon gamma binds to interferon gamma receptor, that results in the recruitment of JAK1 and JAK2, whereas in the case of interferon alpha, it is JAK1 and TYK2. Now, when JAK1 and JAK2 are recruited to the interferon gamma receptor, that results in the phosphorylation of the interferon gamma receptor, that results in the recruitment of STAT1, but not STAT2. And these STAT1s now form homodimers, they in turn go and bind to distinct set of elements and activate the transcription of specific target genes. So, despite of the fact that these cytokine receptors act through Janus kinases and various STAT molecules, there are differences what kind of Janus kinases are recruited to the activated receptor as well as what kind of STATs are activated by these Janus kinases, and this differences is what manifests each cytokine signaling as a different pathway. The STAT binding sites in the promoters of various target genes are also quite distinct. For example, the STAT1 binds to what is called as the interferon stimulated response element, or known as ISREs, and this is the nucleotide sequence to which the STAT1 heterodimer goes and binds. Okay. So, the target sequence for the STAT1 is known as ISRE, and this is the particular sequence to which this binds. Whereas, the binding sequence, the target sequence recognition sequence for the STAT15 transcription factor is known as gas or interferon gamma activated sequence, and this is shown as the consensus sequence of gas is shown here, TTC N3 GAA. And in the case of STAT6, it goes and binds a specific sequence TTC N4 GAA. So, you can see here it is TTC N3 GAA, whereas for STAT6 it is TTC N4 GAA. So, each one of these STATs are activated by different Janus kinases, at which in turn interact with different kinds of cytokine receptors, and these STATs go and bind to distinct response elements in the target genes, and therefore different target genes are activated in response to each one of the STAT molecules, and that is why when you knock out these STAT molecules, you get different kinds of physiological responses, because they go and activate different kinds of target genes. So, what I have now done is I have just drawn some cartoons. These are all taken from the essay by Inces Pathway Central, very nice cartoons just for you to get an understanding how each one of these cytokines activate expression of various target genes. For example, if we take the interferon alpha or beta, they go and bind to the interferon alpha receptor, which is a homodimer, and when interferon gamma binds to the interferon alpha receptor, it results in the activation of the JAK1 and TYK2. The JAK1 and JIK2 then phosphorylate the interferon alpha receptor, and once the interferon alpha receptor is phosphorylated, that now recruits STAT molecules, especially STAT1 and STAT2, and once STAT1 and STAT2 are recruited, they are again phosphorylated by the JAK1 and TYK2, and this phosphorylation of the STAT results in the formation of heterodimer, and many times another transcription factor IRF9 also joins the party, and these together go inside the nucleus and bind to the interferon stimulated response elements, or ISREs, and activate the transcription of specific target genes, and this is how physiological responses for interferon alpha and beta is brought about. So, binding of interferon alpha and beta to the interferon alpha receptor triggers a pathway involving STAT1 and STAT2, and all the target genes which contain these ISREs are now bound by these, and they activate the expression of these genes, leading to specific interferon alpha specific responses. Whereas, if you go to the interferon gamma, in the case of interferon gamma, when the interferon gamma binds interferon gamma receptor, it in the case of interferon alpha it was JAK1 and TYK2, but here it is JAK1 and JAK2, and these two JNS kinases are now recruited to the receptors, and they phosphorylate the receptor, and this phosphorylated receptor now serves as a docking site for STAT1 homodimers, and when these STAT1s are phosphorylated by the JAKs, the phosphorylated STAT1 now forms a homodimer. They go inside the nucleus and bind to interferon gamma or the gas sequence of target genes, and then activates a different set of target genes. So all the genes which contain the interferon gamma activating sequence of the gas motifs now get activated by the STAT1 transcription factor. So in the case of interferon alpha, it was JAK1 and TYK2, here it is JAK1 and JAK2, and there it is STAT1, STAT2 heterodimer, here it is STAT1 homodimers, and the response elements are also quite distinct. So this slide summarizes both the pathways. The interferon gamma primarily activates the JAK2, and through this the STAT1 homodimers are activated, which go and binds to the gas sequences of target genes, whereas in the case of interferon alpha, the STAT1 STAT2 heterodimer is activated, which now goes and binds to ISRE sequences and activates the transcription of target genes. Now the signal transduction pathways are always complicated, and you can see here, in addition to the things, there is also a crosstalk involving PI3 kinase, which is also activated, which then activates protein kinase C, that in turn can phosphorylate STAT, and then it can lead to activation of the STAT. Similarly, the activation of the interferon alpha signaling pathway involving JNS kinases can also go and then activate PI3 kinase, which again results in a different kind of signaling molecule, leading to activation of different kinds of metabolic enzymes. But since our focus is primarily on the gene expression, we will not worry about the events which take place outside the nucleus when these pathways are activated. So we are going to confine ourselves to what are the events which happen inside the nucleus when these signal transduction pathways are activated. Here again another example of growth hormone. We know we need growth hormone for our regular growth. Now how does growth hormone promote growth? Growth hormone binds to a growth hormone receptor, and when growth hormone binds to growth hormone receptor, again a STAT3 and STAT5 are recruited to the phosphorylated receptor, and the JAK2 and JAK2 is the JNS kinase which is involved here, and these STATs again go into the nucleus and bind to growth hormone responsive genes by binding to specific targeting sequences and then activating transcription of these genes. This results in the proliferation of growth or growth of the organism. So the physiological response of growth hormone is mediated by this particular cytokine signaling pathway involving STAT3 and STAT5. The case of interleukin 2, for example, the interleukin 2 is slightly different from the interferon alpha and beta signal transductions we discussed so far. Here the interleukin receptor often is a trimer, consists of alpha, beta and gamma subunits, and when the interleukin 2 binds to these interleukin 2 receptors, again it results in the recruitment of JAK1 and JAK3. You can see here we are taking a different kind of a JAK combination here. JAK1 and JAK3 get activated, they get phosphorylated when the IL-2 docks to the IL-2 receptor, and this JAK now phosphorylate the receptor and then also phosphorylate STAT5, and this STAT5 now forms homodimers, and this STAT5 homodimers now go inside the nucleus and bind to specific sequences and activate the expression of genes, and especially it leads to the production of various cytokines. So you can see different JAK kinases are involved in activation by different cytokines, and different STAT molecules are involved, and different kind of target genes are activated, and this is the signal transduction pathway by which IL-2, interferon alpha, beta, growth hormone, interferon gamma activate specific target genes involving specific JNS kinases and specific STATs. Now very interestingly, the interferon, the IL-2 receptor can exist either as a homodimer involving 2 alpha subunits, or as a heterodimer involving beta and gamma subunits, gamma and C subunits, and it can also have all the 3 subunits together. And it turns out they can exist either in the homo or heterodimers form, and the when you have only the interferon when you have only the alpha subunit homodimers, the affinity of the IL-2 to IL-2 receptor is very, very low. It is something like 10 to the minus 8 molar, and this kind of homodimers are expressed only by activated T cells, and because of their very low affinity they have no function at all. Whereas when they exist as heterodimers, with beta and gamma, gamma and C, the affinity slightly goes up. It is now 10 to the minus 9 molar, and such kind of IL-2 receptor heterodimers are found in resting T cells. The affinity is termed as intermediate. whereas the high affinity intraequivalent receptors involving the heterotrimers have very high affinity, and these are the ones which actually play a very important role, and they are present in the activated T cells. So, this just summarizes the various JAK-STAT pathway. Again, I discussed the growth hormone receptor pathway, I discussed about the interferon alpha receptor pathway, and the interferon gamma pathway, and basically this slide, I do not expect you to understand all these things. The slide is primarily to tell you that in addition to each receptor acting in a specific STAT pathway, you can also see there is a lot of crosstalk. Sometimes the activation of the GPR signaling pathway or receptor tyrosine kinase pathway can also lead to the phosphorylation of tags, and all of them can also together can activate target genes involving a cumulatory effect leading to ultimate activation or repression of various target genes. So, signal transduction pathways are never isolated. So, when we talk about interferon gamma signaling through STATs, it does not mean that other signaling pathways are not activated. Ultimately, a target gene is activated by not only through one particular signaling pathway, but also due to crosstalk between a number of other signal transduction pathways. So, the ultimate expression of a gene depends on signal received from multiple signal transduction pathways, and that is how the sum total of all these events ultimately manifests as the form of a activation or repression of a specific target gene. This again the IL-2 pathway again tells you, in addition to the activation of the STAT5, you can also, there is an activation of NF-kappa B, and you can also see there is a phospholipase C gamma pathway that is involved, resulting in the activation of MAP kinase pathways and activation of the AP1 or the C-Jun and C-Phos, and you can see ultimately the target genes are activated not only by the STATs but also by NF-kappa B, C-Jun, C-Phos, C-MYC, as well as various other transcription factors. So, many a times the activation of a target gene depends not only on one particular signal transduction pathway, but also because of crosstalk between a number of other signal transduction pathway, and simultaneously a number of transcription factors are activated by receiving signals through different signal transduction pathways, leading to very high levels of activation of a particular target gene. How do once the JAK-STAT pathway is activated, how is it terminated? Again, I will not go into details. It is again a very interesting and a lot of network that operates here. It involves two important proteins known as the SHP1 and also the SOX or suppressor cytokine signaling, and when these are activated, it results in the degradation of the STATs involving ubiquitination and proteasome pathway involving a group of proteins belong to the PAS family, but I will not go into the details of how once the JAK-STATs are activated, how this pathway is terminated. Those of you who are interested can read this excellent review by Alexander et al. An annual review of immunology in the 2004 volume 22 page 503 gives you a very good review of how this JAK-STAT pathway is inhibited once it is activated through these molecules. Now, so what we have discussed so far is how cytokines, which are very important molecules, when they dock to cytokine receptors, recruit Janus kinases, and when these Janus kinases are recruited, it results in the activation of the STATs, and different kinds of STATs either form homodimers or heterodimers, and then they go inside the nucleus and bind to specific response elements and activate the transcription of various target genes. Now, the title of this course is Eukaryotic Gene Expression, Basics and Benefits. So, whenever I discuss some of these important topics, I always try to put some information on how understanding the signal transduction pathways or understanding how specific genes are activated by the signal transduction pathways has helped the mankind or has helped to bring about new drugs or new therapeutic agents. For example, I am going to show some couple of slides here to tell you how very important drug molecules have been brought about based on our understanding of the cytokine signaling pathway. A very popular drug known as Enbrel is nothing but it is a monoclonal antibody against the TNF alpha receptor. So, the Enbrel basically is a monoclonal antibody. It prevents TNF interacting with TNF receptor, and therefore, the TNF signaling pathway is not activated, and this Enbrel is a very important drug used in the treatment of rheumatoid arthritis as well as an autoimmune disease called Crohn's disease. So, you can see how molecules which interfere with the signal transduction or the cytokine signaling pathways can be very potent drug molecules. Another example, we have molecule interferon A, which is nothing but interferon A2B, which is very widely used in the treatment of hepatitis C as well as certain melanomas. Epogen is nothing but recombinant erythropoietin. Erythropoietin is a very potent stimulator of red blood cell production. So, if you have anemia or if you are undergoing kidney failure patients who cannot produce erythropoietin, erythropoietin is given to stimulate red blood cell production. So, it is a very important activator of erythropoiesis. All of them act through the JAK-STAT signaling pathway. Similarly, Actimmune is again nothing but interferon gamma 1 beta, again used in the treatment of chronic granulomatous disease as well as osteoporosis. We have another drug Neupogen, which is nothing but recombinant granulocyte colony stimulating factor. It stimulates the production of neutrophils, reduction in the infection in cancer patients who undergo chemotherapy. Cancer patients who undergo chemotherapy, because the immune cells are also getting destroyed, they are very susceptible to various infections. So, such patients are given GCSF actually to promote the production of the immune cells or the T cells and B cells, so that they can become less susceptible to various infections. So, GCSF is very important molecule there. Similarly, GM-CSF is marketed in the trade name leucine, which actually stimulates the production of myeloid cells after bone marrow transplantation. Numega or Neulasta is nothing but recombinant interleukin 11. It actually is used in the stimulation of production of platelets, which helps in blood clotting. So, what we have discussed so far is cytokine signaling is very important, and in the cytokine family is a huge group of molecules, of diverse molecules ranging from interferons, interleukins, erythropoietin, GM-CSF, growth factor, growth hormone, and so on and so forth. And many of them actually act through non-receptor tyrosine kinases, and when these molecules bind to the cell surface receptor, and especially they recruit specific Janus kinases which interact with molecules called STATs, and these STATs, which again are diverse type of molecules having non-overlapping functions, they in turn go and bind to specific target elements of target genes, activate or repress transcription of various genes. I have now listed a number of references here, again taken from a very important website promoted by the SA Biosciences called Pathway Central, and if you read some of these references, it gives you a much more appreciation of how important the cytokine signaling pathway in controlling a number of diseases. For example, the JAK-STAT pathway in rheumatoid arthritis gives an excellent review of how, by understanding the JAK-STAT pathway and how these pathways activate in rheumatoid arthritis, people are trying to develop various drugs for treatments of arthritis. Cytokine STATs and liver disease, again correlating how basic research can be used for development of various drug molecules. Again, inhibition of interferon stimulate JAK-STAT signaling by tick-borne flavivirus and identification of NF1 as interferon antagonist. How JAK-STAT pathway is activated in specific viral infections, and how you can actually use how viruses actually interfere with this interferon signaling pathway and then evade the immune system so that they can survive inside the cells. The role of JAK-STAT signaling in cancer, as well as the last two actually tell you how JAK-STAT pathway plays a very important role in number of cancers. A few more references basically to give you an idea how important are these, especially this article discusses about how the JAK-STAT pathway is actually inhibited by the SOX1 pathway. I would like to acknowledge that many of those colorful pathway slides I have shown you so far have all been taken from the SA Biosciences Pathway Central, a beautiful website for understanding a number of signal transduction pathways, and you can just click on or cut paste this web link and you can go, and then you can just have to log on, put your username and give a password. It is a free website. People, any academic researcher can actually use this to understand various signaling pathways, and very colorful slides and very colorful illustrations are given to understand how various signaling molecules activate distinct signal transduction pathways leading to activation or repression of specific target genes. So, I think we will stop here. So, what we have discussed so far in this series of signal transduction lectures, about 5 lectures from lecture 15 to lecture 20, we basically covered one aspect of signal transduction pathways. We primarily talked about molecules which interact with G protein coupled receptors, which activate through receptor tyrosine kinases, and those which now activate through the JAK-STAT pathway. There are many other membrane receptor molecules, there are many other pathways by which various membrane receptors act through, but it is not possible to cover all these things. So, I have just given you a very brief idea of how molecules which cannot enter the cell can bind to specific membrane receptors, and these 3 examples tell you how binding of these molecules to membrane receptors can transduce signals either through MAP kinase signaling or through specific protein kinases like protein kinase C or protein kinase A, or in the last case we have discussed today the JAK-STAT pathway, ultimately results in the activation or repression of specific target genes manifesting in the form of distinct physiological responses. With this, we have completed the signaling and regulation of gene expression by molecules which bind to cell surface receptors. But I strongly encourage you to go and read up a few more interesting examples like transforming growth factor beta, erythropoietin, hypoxia inducing factor. These are all wonderful examples to understand how this membrane signaling works and how each one of these receptors bring out distinct physiological responses by activating different kind of protein kinases and so on and so forth. And they all have very important biomedical relevance. What we will do in the next class, we will now move inside the cell. We will now talk about molecules which actually diffuse through the cell membrane but bind to specific intracellular receptors, and then when this signaling molecule or the hormone receptor complex then goes inside the nucleus and activates the transcription. So far we remained in the outside the cell phase, talked about molecules which interact with membrane receptors on the cell surface. From next class onwards we are going to move inside the cell and talk about molecules which go inside the cell, interact with specific intracellular receptors, and then activate or repress the transcription of specific target genes. I will stop here.